My name is Ikenna Kingsley Uchendu. The title of my talk is Significance of Cytokines in the Diagnosis and Prognosis of Oncological Diseases. This is the outline of my talk. We'll quickly look at a brief background. Then we'll look at cytokine actions within tumor microenvironment. Then we'll go into looking at significance of selected cytokines in the diagnosis and prognosis of oncological diseases. So we'll be looking at 30 cytokines, and then we'll go on to look at published evidence for specific cytokine effects in various cancer types. Then we'll conclude. Background. Tumor markers are tumor-associated proteins in cancer patients that are clinically significant. Despite the fact that some tumor markers are frequently utilized, they neither show a high level of diagnostic accuracy in all tumor types. For this reason, promising indicators for cancer staging, prognosis, and therapy response include cytokines, serum cytokines. In fact, it has been suggested that circulating cytokine studies in conjunction with cancer-specific biomarkers can facilitate and enhance cancer diagnosis and prognosis. Now, this is the role or actions of cytokines within a tumor microenvironment. Within a tumor microenvironment, there are many cell types. There are cancer cells, or we call them tumor cells. There are stromal cells, and there are immune cells. These cells interact with one another, and their interaction is modulated or controlled by cytokines. And these cells themselves also produce or release cytokines. Now, these cytokines may play one of these two roles. They may promote tumorigenesis or cause initiation and proliferation of tumor cells themselves. They can cause immune suppression in the patient. They can cause drug resistance or medication resistance in that patient. They can also promote angiogenesis and eventually metastasis of tumor cells. Or they may go on to suppress tumorigenesis. So they can play these two roles: promote tumor progression or suppress tumor progression. So for this reason, these cytokines are very, very important. When they find their way into the blood, they can be used to diagnose a particular kind of tumor. The balance of tumor promoting and tumor suppressing components of the immune system plays a crucial role in cancer progression and treatment response. Cytokines are key players in immune cell signaling. So now we're going to be looking at 30 different cytokines that have significant roles in the diagnosis and prognosis of oncological diseases. So the first three are interleukin-1, which has two isoform, two subtypes, interleukin-1 alpha and interleukin-1 beta. Now, interleukin-1 production locally affects tumor growth and metastasis, either directly through proliferative effects or by encouraging inflammatory and angiogenic pathways in host cells. Now, interleukin-1 alpha and interleukin-1 beta, both are subtypes of interleukin-1 and both attach to the same receptors, but they undergo through different processing steps. Now, a little bit of the difference between the two of the subtypes. Interleukin-1 alpha is active in its precursor form, while interleukin-1 beta is active in its mature form. That's one of the two differences between the two of them. But they both bind to the same receptor and they both promote tumor growth and metastasis. So let's look at interleukin-1 receptor antagonists, LI1RA and interleukin-2. Now, as it sounds, the name implies interleukin-1 receptor antagonist. This interleukin opposes the effect of interleukin-1. So the same goes with interleukin-2. Now, let's talk about the two of them. Interleukin-2 and interleukin-1 receptor antagonists are both tumor-suppressing cytokines. Both are differentially expressed in the serum of advanced pancreatic cancer patients with disease control and patients with progressive disease after treatment with FOLFIRINOX chemotherapy. So to throw a little more light on this, now a research done by Van den Siege et al. 2021, they sampled 88 patients with advanced pancreatic cancer, And these patients were divided into two groups. One of the group were named disease control group. The other group were named progressive disease control group. Now, the progressive disease control group were treated with sulfonylureas chemotherapy over time. At the end of the treatment, already some blood samples for the assay or measurement of interleukin 2 and interleukin 1 receptor antagonists were taken before the treatment and after the treatment. Now, at the end of the research, it was discovered that interleukin 2 was the detection rate of interleukin 2 was higher in the disease control group than in the progressive disease group. But at the end of the chemotherapy, this was the reverse was the case. But for interleukin 1 receptor antagonist, at the end of the chemotherapy, the detection rate of interleukin 1 receptor antagonist was higher in the disease control group than the progressive disease group. So you can see there was an interchange of the two. For this reason, they concluded that the two interleukins or cytokines are differentially expressed in the serum of these patients with the disease mentioned, the tumor mentioned. So interleukin 3 and interleukin 4. Interleukin 3 plays a potential role in the development of tumor blood vessels, thus angiogenesis. This is because each receptors are located in the vascular system. This underscores the necessity of using certain monoclonal antibodies to target the interleukin 3 receptor. Recent research in colorectal cancer has confirmed the relationship between interleukin 4 and the aberrant STAT6 activation. The STAT6 stands for signal transducers and activators of transcription 6, and STAT6 is known to regulate signal transduction and encourages cancer spreading activities. Interleukin 5 and interleukin 6. Interleukin 5 is one of the several inflammatory cytokines that are associated or accompanied with the emergence of bladder cancer, making them prospective and significant novel target molecules for the control of bladder cancer migration and invasion. While interleukin 6 has been implicated in the development of malignant tumors like colon and gastric cancers, pancreatic cancers, and lung cancer. And this is by the effect of the action of increased STAT3. And STAT3's activation is caused by excessive production of interleukin 6. Interleukin 7 and interleukin 8. Interleukin 7 expression levels were discovered to be higher in colorectal cancer patients, in contrast to the control group. And this was found to be related with metastatic diseases and tumor location in this patient. While interleukin 8 is involved in inflammatory-related clinical disorders. Fibroblasts, endothelial cells, and tumor cells are its primary secretor. Interleukin 9 and interleukin 10. Significant importance of interleukin 9 in the tumor microenvironment of colon carcinoma is suggested by the fact that its expression is adversely connected with tumor node metastasis staging, DOC staging, and lymph node metastasis, but positively correlated with favorable prognosis. So for interleukin 10, increased systemic interleukin 10 concentrations are associated with advanced stages of cancer, but higher level of interleukin 10 concentrations are frequently associated with poor prognosis. Tissue necrosis factor and interleukin 10. Small levels of tissue necrosis factor are produced by malignant cells on a regular basis, which makes blood vessels more permeable. In a lung cancer model, this resulted in pleural effusion being promoted. TNF aids in the maintenance of an inflammatory environment in existing tumors. For interleukin 13, interleukin suppression has shown that interleukin 13 expression levels are inversely connected to the estrogen receptor and progesterone receptor, suggesting a potential role in the severity of breast tumors with estrogen receptor. Interleukin 13 is therefore being examined as a possible breast cancer prognosis indicator. Interleukin 15 and interferon-inducible protein 10. Interleukin 15 is a cytokine that preferentially stimulates the CD8 T cells and natural killer cells activation. Interleukin 15 and cytokine, Interleukin-15 is a cytokine that preferentially stimulates CD8 T cells and natural killer cells activation, proliferation, and cytotoxic activity. Not surprisingly, this functional activity of interleukin-15 translates to enhanced anti-tumor responses in multiple tumor models. Thus, interleukin-15 is a potential cytokine for the therapy of hematological and solid malignancies. Multiple myeloma, malignant melanoma, ovarian cancer, basal cell carcinoma, and other advanced women's cancers have been linked to elevated expression of interferon-inducible protein 10 and its related receptor CXCR3. Macrophage inflammatory protein 1, which has two isoforms, two subtypes, MIP1 alpha and MIP1 beta. Now, MIP1 is one of the members of the MIP-CC chemokine subfamily. MIP1 alpha and MIP1 beta levels of expression were observed to be higher in breast cancer patients in this study by Lee et al. 2017 compared to healthy control participants, indicating that MIP1 alpha and MIP1 beta play a role in the pathophysiology and progression of breast cancer. MCP1 and MCP3. MCP stands for monocyte chemoattractant protein 1 and monocyte chemoattractant protein 2, 3. MCP1 is a powerful chemokine that attracts blood monocytes to tumors and sites of inflammatory responses. It was reported that compared to patients with modest producers, those receiving surgical resection have a higher or much greater survival rate. Therefore, patients with higher MCP1 levels have better prognosis. MCP3 is essential for the development of tumors. MCP3 is reported to promote tumor growth by aiding in development of the tumor microenvironment and enabling tumor invasion and metastasis. RANTES and VEGF. RANTES stands for regulated upon activation, normal T cell expressed and secreted. And it is also known as CCL5, chemokine ligand 5. RANTES is chemotactic for T cell, eosinophil and basophils. It plays an important role in recruiting leukocytes into inflammatory sites. Zhou et al. In 2017 evaluated the expression of CCL5 in tumor tissues from women with serious ovarian cancer. Their findings show that RANTES may increase drug resistance via cellular paracrine or autocrine pathways. For VEGF, VEGF stands for vascular endothelial growth factor, which is also known as vascular permeability factor, VPF. It was originally described as the endothelial cell-specific mitogen. The evaluation of VEGF production could be helpful in predicting the spread of colorectal cancer as a result of VEGF's recognition as a key angiogenic agent in colon carcinoma. Interleukin-12 P450 and MDC. Interleukin-12 P450 is part of the bioactive cytokines interleukin-12 and interleukin-23, and these interleukins are believed to be central to the CD4 T cell response and key driver of cell-mediated immunity. Interleukin-12 P450 levels were assessed by ELISA in the sera of 91 patients with colorectal cancer who had not previously received treatment. Now, with the development of colorectal cancer, the levels of circulating interleukin-12 P450 substantially reduced. MDC, macrophage-derived chemokine, is thought to operate as a chemotactic for dendritic cells, natural killer cells, and T lymphocytes. Elevated MDC gene expression in tumor tissues is suggested to possibly serve as a diagnostic indicator for lung cancer that would improve prognosis. Interleukin-12 P70 and soluble cluster of differentiation 40L. Cytokine interleukin-12 P70 is produced by dendritic cells. Dendritic cells' capacity to activate T cells is strengthened by increased levels of interleukin-12 P70. In an investigation, interleukin-12 P70 along with interleukin-6 and interleukin-8 were discovered to be the three crucial markers that might better predict tumor metastasis. Soluble CD450L is released by activated T lymphocyte and platelets. It has previously been reported that SCDL40L is produced at high levels in lung and nasopharyngeal carcinoma lesions. Cancer patients also manifest an elevated propensity for platelet activation. According to Angelo et al., patients with neoplasia and greater levels of circulating SCD40, and it is possible, sorry, according to Angelo et al., patients with neoplasia had greater levels of circulating CD40L, and it is also possible that CD40L may play a predictive role in this patient's outcome. PDGF AA and PGDF AB or PGDF BB. Mesenchymal cells and a few other cell types are stimulated to grow, survive, and move by the PDGF isoforms. They play crucial roles in adult tissue homeostasis regulation as well as embryonic development. Both non-malignant conditions marked by excessive cell proliferation and some malignant disorders are linked to an overreactive or active PDGF signaling. Overactive PDGF signaling has been linked to non-malignant disorders according to research by Heading 2013. Interleukin 17. Interleukin 17, which is a pro-inflammatory cytokine generated by cells with CD4 T helper 17 and CD8 T cytotoxic 17. Interleukin 17 cytokines either directly damage tumor cells or indirectly do so by impairing the patient's anti-tumor response and creating microenvironment changes that worsen the disease invasive and metastatic features. Increased expression of cells that produce interleukin 17 in the tumor microenvironment of mammary gland tumors is associated with a poor prognosis for staging, surviving in general, and survival without disease. Now, these are published evidence for specific cytokine effect in various cancer types. So on the left is the cancer types, and on the right are the inflammatory mediators that the researchers reported on. Conclusion. It is outside the scope of this presentation to provide a comprehensive assessment of individual cytokines because of time. This summary approach provides a necessary simplification of the scenario, leaving out various cytokine activities and interaction. Thank you.